Well, thank you, you guys, for joining us today. This is our fourth expert series talk that we've started. I am super excited to present Dr. Ari Martinez. So he is an ecologist whose research interest lies at the intersections of behavioral and community ecology. For example, how does sociality among species influence the organization of animal communities? To answer these research questions, along with others, he has spent over 10 years living and working in Latin America using birds of the Amazon rainforest as a study system. In order to do so, he has slept in everything from beds, a tent, a hammock, and on village sidewalks, and traveled in everything from an ox cart to a motorcycle, a dugout canoe, to a lodging truck. In 2018, with funding from National Geographic, he organized the research teams to recensus an entire Amazonian bird community after an initial census 35 years earlier, and which included three of the original team members. The talk today is about the challenges of serving Amazonian birds communities and the results of this particular recensus. I also want to include that, Ari is an amazing professor at Cal State Long Beach. I definitely took his ornithology class my first semester as a graduate student, and I definitely learned a lot from his class. And I would, every time after taking his class, I would go to work and I would talk to my coworkers about his class, everything I learned. So I'm pretty sure not only I learned a lot, but a lot from my coworkers have learned a lot from taking his class. So with that, I would take it on to Ari.
Anita, well, thank you very much for that wonderful introduction. And I would just like to say that some of the things I try to emphasize in that class carry over to what I want to talk about today. And one of those big themes is thinking about natural history and the importance of observation when we approach science. But for this larger group, I would just like to say that having some kind of development of being observant in nature is so important, and we can learn everything even from Southern California just by being very observant. So whether it doesn't matter, you don't have to be an ecologist to appreciate nature. And so just like I talk about my class, if you're observant, you don't always need to watch planet Earth to appreciate nature. You can go out your front door and see California towhees and see migrant birds falling out near you during fall and spring. And so there's a lot to learn from that. So with some of that being said, how does an Amazonian forest bird community change after 3 decades? It's kind of one of the main questions we wanted to ask, and this talk is going to talk about that experience and some of the challenges along with it. And so when we think of why would we ask this kind of question, well, as you know, how communities change through time is something I think that, well, as ecologists, we want to understand. And for the general public and for society at large, we want to understand how communities are changing through time, especially now over these trying times where a lot of us are worried about global change. And so to understand how global change is affecting communities, we need to understand natural dynamics of communities and then understand how those natural dynamics are shifting. And the thing is that just learning how communities change through time. So how do the number of species in a community, how do their abundances change year to year? That is a challenging question in any system. And in fact, here's one of the few data slides I'm going to present. But if we look at this, these are data slides that go from 30 years data sets for interesting sampling phytoplankton communities, or to look at desert plants, or to marine invertebrates. And you see these changes up and down from year to year. And so what you're looking at are on the x-axis just the number of years, and you can see they expand anywhere from 15 to 30-year data sets. And on the y-axis, don't worry about it, but there's all different metrics of abundance. And so what you can see there is that each of these different boxes represent a different species. And what they've done is they've measured each of these species in the same way, and they've done it for a long period of time for these different communities. So the interesting thing here is that when you look at these, for phytoplankton, to do that, you go to the same places and you maybe use these, you take out nets and you haul nets and you look at the at the number of individuals of these different species, and you go to the same places and you do that. Or for desert plants, you can set down what are called quadrats, these square kind of square meter kind of frames, and you plant them down in the same places year after year, and you count the number of plants you see. Same thing for marine intertidal invertebrates. You go to the intertidal zone, and you go to the same places year after year, and you plant these things down, you count all the different species. So for a lot of different types of biological communities, there's standard ways to do that. And so that's why you end up with these long-term data sets where you can go year to year and see what's been done. Okay, well, in rainforest, it's very challenging in the sense that you have obviously a very diverse community. So instead of looking at 10 species, you're looking at close to 400. And on top of that is the fact that up till about 30 years ago, we didn't even know how to identify a lot of these species. Things would call and they would vocalize in the rainforest in terms of birds. And a lot of times people didn't know what they were. So that's why when in 1990, John Terborg, along with his lab and a famous birder called Ted Parker, they actually made a plot instead of a meter quadrat. They would put down an entire 1 kilometer grid to survey all the birds in a parcel of tropical rainforest. And so it was the first time anyone had described the structure and Amazonia organization of an entire bird community, because we finally knew enough about all the different types of birds that in terms of IDing them, that we could identify them. And this happened in southeastern Peru. You can see that the arrow there shows, and that dot shows exactly where it was. And so this was a huge accomplishment back then, but it took a lot of manpower, a lot of people power and person hours, and a lot of people, and it took a lot of time. This was over 3 months that they did a survey, but they complemented it with 2 years of misnetting data and following flocks from some of the graduate student theses that were in the work, that were in part of this project. And so it took a lot of time. And we're going to go here to this to Manu National Park and understand a little bit what some of the challenges of how to go about of why this was done, and how to repeat it, and how this can help inform decisions in the future. So here's Manu National Park. It's in a very remote part of the Amazon. It's one of the few places that still doesn't have any kind of human impact other than pre-Columbian societies that are still living there. In other words, uncontacted tribes. And so you can see that it's incredibly vast. There's a lot of there's a lot of undisturbed rainforest. There's no logging, no mining in a lot of parts of the park. And it's a beautiful place, but it's also very remote. It takes a very long time to get there. And just to give you a sense of the vastness, that's one of the boats, for instance, that takes you there. And it literally takes you at this speed in 90% humidity with very little wind. You're puttering along on one of these boats, and it takes literally 2 days to go along these meandering, winding rivers. This is the Manu River, and it takes literally up to 2 days to get there by boat. And then before you even get on the boat, you need to drive up and over the Andes 1 day by car. So it takes up to 3 days to get to the field site that we're going to go to understand where we find this plot that they originally surveyed, and to be able to look at this, to look at these birds and be able to recensus this area. So if we go, here's the actual field station. There's an eventual endpoint of where we're going. And here are some of the birds, for instance, that we're going to eventually survey. But the crazy thing about surveying all these birds is that to survey all these birds, again, like the original study, you need a parcel that's about 100 hectares, which is about 100 acres to you. Well, it's about 200 acres to those of us who don't deal with metric systems. So it's a huge plot, and it's based on Cocha Cashew. This is the Cocha. A Cocha is an oxbow lake that has been that is pinched off from the river, and then it forms this oxbow, what's called this oxbow. It's one of these original ones here, and eventually the river punches through and abandons that. So this is one of the original bends in the river as the rivers shift and meander through time. And so there's 245 resident bird species that were censused in this plot, 319 total when you count all the migrants and transient bird species. And the thing is that in today's age, what I want to kind of emphasize, especially for those of you, the general public, we think of all these things that get reported about how we do science because they're very attractive in terms of like, well, thinking about technology. And so people try and storytell about how technology is so important to tackling conservation, and people like to use it a lot and weave all the technology into stories, and they are in fact helpful. For instance, conservation drones, people have used these for a lot of different types of conservation applications. In fact, in the tropics, people have used them to do transects to survey the nests of orangutans in Malaysia, for instance. And then you've got, of course, automated recorders that look at that record sound and soundscapes over time. So people leave them out there, and then people can apply machine learning and artificial intelligence to ID whatever it is that's specific to the certain species they want to detect. Or in fact, if people want to set them out there so that they can detect chainsaws, and then they can hear legal logging in different parts of the forest. And then, for instance, we know about remote sensing that can look at different types of signatures through satellite imagery, and that can tell us something about the structure of forests or the way we can evaluate carbon stocks in the forest. And then lastly, there's these MODIS systems that actually are these long-term systems that are set up that when you can put little transmitters on birds or animals, and you can look at migration over time by seeing whether when they fall within the detection grid of these MODIS, different MODIS towers, we can see how things migrate long-term over different areas. So we have all these different technologies that can help us in conservation and that are hugely important. So the question is, with all this technology, we can think, 30 years ago, we went through and we could ID these birds because people learned about the natural history and they learned by ear, right? So those different individuals that were the people that were that published that original paper on the structure and organization of the bird community, they literally went through the forest day after day and counted what they heard and saw through natural history and did these different techniques like spot mapping that I'll get into. So how can we evaluate Amazonian bird communities and how do they change through time and how do we use it with all this technology? Well, the fact is we need to use multiple techniques at once to estimate population densities for all these species. And in fact, none of those technologies help us. My point here is that in this sense, what they did 30 years ago is literally what we have to do today. Obviously, we want to, well, not obviously, but for those who maybe in the general public, one thing is if we want to compare changes in communities through time, we want to use the same methodologies that we used back then and use them now. But then the other thing to think about here is, in fact, that with all these fancy technologies, what it comes down to census things like the diversity of Amazonian birds or diversity of a lot of taxonomic groups, you need to know the natural history. A lot of these technologies still can't help you. And to know the natural history, you have to learn the birds by call, by knowing all their vocalizations and be able to identify them visually. And so once you do that, we can do a lot of different techniques at once to census all these different types of bird species. And so we're going to use a combination of all these different types of methodologies to census a bird community like they did back 30 years ago. And the reason we need to use all these different techniques is because, in fact, there's so many diverse types of birds that do different things that one technique can't apply to all of them because it doesn't accurately census all the different bird species. So in these other cases that I showed you, where you had a desert plant community, you can put out on a quadrat, and all these plants, you can detect all the plants that are growing in the soil. Or with phytoplankton, you sample phytoplankton all the same way. Birds, you can't, because some birds, they have larger territories. Some have these social organizations that don't allow us to census them in the same way. And as a result, we need to look at these different techniques, and we need to apply individuals that can use these different techniques and then bring it all together to look at the different densities of these species and see how the different densities now compare to back 30 years ago. So this is very daunting. And in fact, I think John Terborg, one of the original authors on the paper, who's my academic grandfather, in other words, my academic advisor, that's his advisor. And I think when we originally decided to do the census and we were trying to get people together to do it, he said, if we're going to get a bunch of people together, and his recommendation was that for anyone who wanted to do the spot mapping aspect of the work, they had to know probably on the equivalent of 1,800 different vocalizations. When you consider the 300 species by the 5 or 6 vocalizations they all know, that would not be the qualification that you would need in order to be able to efficiently go through and spot map all the different species as you detect them walking through in the morning. Okay. So again, this is saying that even in spite of all these technologies, we need good old fashioned powers of observation and good old natural history skills. And it's humans who have to go out and do this work. So for instance, why do we need all these techniques? Well, you have these things like puffbirds. Here is Bucco capensis. I don't even remember what the common name is. I think it's chestnut-capped puffbird, which is a very skulking silent bird. And so you need things like mist nets to be able to capture and ID these things because they're often not very vocal. You have things like antbirds, obligate antbirds that literally follow army ants. So again, following through and census thing by ear might not necessarily be the way they go because they're very patchy in what they do. And then you have things like puffbirds. These guys will never will catch in nets. These are in the mid-story, but they're very vocal. So you can catch them with spot mapping. And then you have trogons, same thing, very vocal. And so again, something where you can hear by ear. And so you can count them by spot mapping. They're in the mid-story. And so you look at all these birds and then these different types of things. And to collect abundance, you have things like spot mapping. We're going to do mist nets to capture birds. We're going to follow flocks. We're going to do group counts of parrots on the river. And then we're going to do colony counts of things like oropendulas. So the single species counts, for instance, like colony and group counts, what we did is we'd go out to the river and literally count the groups as they'd fly over. And for all the species of parrots, we would actually count the number of individuals of each parrot group. And so then when we heard the parrots in the forest, we could estimate if we heard a certain species, like we heard, if we heard a chestnut-fronted macaw, and we obviously counted the number of groups that came over the river, and we knew that our group size was on average about 6 individuals, we could then say, anytime we heard an individual in the forest, that the group size would all be about 6. Same thing with things like yellow-rumped caciques, we would do the same thing, where if we saw groups of 10 flying over, 15 here, we'd get a mean. And so then when we census them in the forest, or get the colony, we can get a good sense for the size of the colonies. So, and a lot of times we have to go out in the middle of the river and census them in late afternoons. And then of course, the other thing we might do is mist nets, where we'd set up mist nets early in the morning. And this is like 5:30 in the morning. Here's Renato, Peruvian biologist, censusing birds, pulling them out of nets. And here's Blaine Carnes, who was setting up the banding station in the early morning. And here's what birds look like when you catch them in the net. And you pull them out of the net, they don't get hurt. You pull them out the net like every half an hour, and you can get these. Here's Blaine with a bunch of birds around his neck. Each one of these bird bags has a bird in it. And you can process these birds, put bands on them, measure them, and get a lot of data about their characteristics in terms of what we call morphometric data, their wing length and their body, their tail length, their tarsus length, bill length, get their body weight to get a sense of their condition. But the other main thing, in order to extrapolate abundances for certain species, like these cryptic species, you can get capture rates. In other words, the number of birds that capture in nets per unit time, and you can extrapolate an abundance for a lot of these cryptic species. And then, of course, here's a sampling of some of the birds you'd capture in the nets. So you have like a mannequin below. You have another puffbird on the upper left-hand corner. You have a tanager on the far right. And then below again, you have what's called a type of trogon. So lots of different birds and lots of different histories. And then once in a while, you can catch some crazy things like pteroglossus, which are a type of toucanet, or a type of what's called an arasari, which is a small type of toucan. Okay, so we can get lots of different things, and these guys are rare to catch in nets, but once in a while you can get them. So the main type of technique that we did was a thing called spot mapping, where you'd walk through these trailed grid systems within the forest, and every time you hear a bird, there's stakes in the forest, you get a distance and direction from a reference point, and you can get these different clouds of points that correspond to the different individuals of each species. And by doing that, you could map out the different territories eventually over time, over 3 months, and eventually get the different amount of individuals of each species that have different territories. And therefore, by having the number of territories per unit area, you can extrapolate and get the densities over of within the plot of each species, and compare that to the densities that were there 30 years ago. And here you have my advisor in the left-hand corner. There's me. And then you have Tomas Valky and Jorge Novoa, who are colleagues of mine in Peru, who were collaborating with us on this project. And so it looks really daunting because this is what it looks like. And you're like, where are all the birds? Well, they're calling, right? The main thing to think about is the vocalizations are the main way we get some of these birds. And so when we think of that, when we do that through time and walk through all these forests, here's one of the original maps of a bird, of a plumbeous antbird. And here are the maps that they developed of all the spot maps of all the points they detected by walking through every morning of all the different of all the different individuals, of all the different of all the different territories in here. And so what they could do is by counter calling, hearing simultaneous birds at the same time, they would draw these boundaries. And that's how you would draw the boundaries of these territories. So then what you could do is, this is what the map looked like that they drew from the original from the original paper. And this gave us the number of individual territories of this species, which gave us a density, which is a measure of abundance of that species. And so now we did the same thing back in 2018 because we redigitized all those original maps, because they literally had all those drone maps, my advisor, from 30 years ago, and we had someone redigitize them. And then we can compare them to now. And if you can look, so there's variance here. But when you see this, it's kind of eerie that when you look, the overall abundances are pretty similar. And so this was a different person doing these maps. So they didn't have the bias of what they thought they had 30 years ago. And so this was a different person drawing these maps. And so we can see is the abundance of this species is eerily similar for this species of this Columbia's hamper. And so the one thing I should talk to you before is, when I've talked about these maps, we've talked about you noting about the vocalizations. And what I want to convey to you is that, and this is what I convey in my classes all the time, is that when you become observant at nature, you understand the nuances of diversity in the subtleties of the behaviors and the vocalizations of species. So while we all get stunned and we all ooh and ah because we want to see colorful birds when we think of the tropics, right? There's these different ways of understanding diversity that it's just not that we can use our other senses to understand. And one of those is the vocal form. So here, for instance, is a potu. So I just want to give you a sense of how, if you think things are diverse in the way they look in a rainforest, they're also diverse in the way they sound. So listen to some of the sounds from the rainforest. Oh, sorry. It's kind of a haunting sound, right? Because we also have some of these at night. Here is a musician wren. One of the most now when I've played this bird, I've had field assistants say, "Who's also in the forest with us?" I'm saying, "No one's in the forest." Well, they someone's singing, whistling, and like, no, that's a bird, and this is the musician wren that are in groups. Almost like playing a flute. Okay, here's a cacique. I mean, crested or pendula, sorry. And here, this is from a different site, but here's a capuchin bird. They accompany mosquito trying to bite me. Here's a Nasica longirostrus. long-billed wood creeper. Lawrence's thrush. This bird mimics a bunch of other birds, common birds. Almost sounds like that rattle is a very distinct call of this flocking bird. Almost sounds like a machine gun rattle. And that's it, doing all these little calls, all these different vocalizations that this bird does when it's agitated. And this bird gives alarm calls. So you can hear that there's a lot of diversity in terms of the way things sound. And so that's one of the axes of diversity that I think that gets underlooked. And so you can hear and appreciate the way that things are just, that you can observe things not just by your eyes, but also your ears. And then even if you're studying trees, you can smell the bark, right, to understand differences between trees. So you can use a lot of your different senses when you're in the rainforest. Okay, so what do the results show? So I want to show you, for instance, these heat maps that just give you a sense for, based upon the spot maps, of how similar the distribution of the bird abundances are between time points. So remember, I talked about, we went through and on these grids, we put a point down every time we detected an individual, and we got these territories. But another way to do this is we had all the points where we detected the species over 3 months for all of us doing the spot mapping on the plot. And then what we could do is we did a technique what's called a kernel density estimation, which is you basically develop a heat map based upon the concentration of points for each species. So we could put all the points of a species together, and we could generate a heat map of where it's more common and where it's less common. And then we could compare those heat maps between the two time periods to see how much there's been a shift in terms of where you see the species on the plot over time. And if they're pretty similar, it tells us most likely those territories are also pretty similar over time. And so here's an example, if you look, of 6 of these species. And what you can see is how eerily similar. The yellow means it's more concentrated, red is absent. And what you can see is the concentration of the species when you put all these points of all these territories together. For these 6 species, it's almost identical. This is after 35 years. And so it suggests that for a lot of these species, that the distributions and the territories are likely very similar and are very stable through time. So here, for instance, we have one territory of Automolus rupilatus, a type of foliage gleaner. It's literally this one territory back in 1982, which when they did the survey, and back in now in 2018, we literally resurveyed the same place. That single territory is there after 35 years of one bird. Okay, now here's obviously multiple territories. This is contracted clearly, but here you see it's almost identical for this wood creeper, almost identical for this antbird, almost identical for this tinamou, and almost identical for this jacamar. So very different types of birds from different families, and almost all of them are very similar. And when you look at these densities, this is a way we can look. So this is the only data slide I have from the graph. It's changed a little bit with our different analyses and our different struggles with reviewers from back and forth struggle from trying to get this published. And when you can look at is the density. If you look here, we're trying to predict the density of the birds of each point is a different species, and we're trying to predict the densities from 2018 by looking at the original densities from 1982. So this black line would be a 1-to-1 line. In other words, this black line says, if I had a density of 10 individuals in 1982 and I predicted 10 individuals in 2018, if it was also 10, it would be 1-to-1. And if you did that for all the densities of the birds, you'd have a 1-to-1 line. So if most of these points were below this line, it would suggest that most of the abundances have decreased from 1982 to 2018. If most of them are above the line, it would suggest that they've increased. And what we typically tend to see overall is that the densities haven't changed. The dashed line was what we see, and the gray area is kind of like our confidence in our estimate. And what it suggests is overall, while there's lots of variance of things above and below, overall the densities haven't really changed that much. Biomass is when we calculate overall biomass for the different grams per hectare for the different species as we go from 1982 to 2018. This has changed a bit. This is much more similar to 1-to-1, We don't see much change with our newer analysis. And so biomasses haven't changed a little bit. They show a little bit of a decrease, although some, if you look at the estimate, the larger individual species, if we have a larger sampling, if our sampling methodologies show more uncertainty, because the larger sized birds tend to be birds in groups that aren't really, aren't as confident in our estimate, like things that wander around the rainforest, like big things like these types of you don't want to call them turkeys, but they're called curassows. They walk around in groups, and if you don't see or hear a group over a certain time period, the estimate can be very variable, and that's what can lead to big differences in biomass. And so the one thing I want to say is that overall, our take-home message so far is that there are variants between different types of groups, but overall, and there are increases and decreases in certain groups, but most groups, when we look at the different types of feeding guilds and we look at the overall amount of birds and their densities, they tend to be similar. And other studies have been showing, in fact, that it's in different regions of the Amazon that are looking at tropical birds, one study in Panama, one in Ecuador, and one study in Manaus in Brazil, is that they've been showing declines in remote regions of rainforest that haven't had large scale impacts of human activity, where there's been habitat change or resource extraction. And so they're still seeing declines in areas that haven't been affected by things, and they're pointing to potential changes in precipitation patterns. And those precipitation patterns the question is, are those precipitation patterns changing the resource abundance, and is the driver of that global change? Well, we're not seeing that effect here because, in fact, we do have some data that shows the precipitation patterns in the areas most close, the areas that are closest to this site where we have measurements of precipitation haven't tended to change. And so we see that these areas, in fact, this area in particular, hasn't shown a lot of change, which is amazing from the point of view from ecology, the dynamics of populations and communities changing through time. We don't see much change, which is pretty incredible. But the other thing is, does this site then serve as biodiversity refugia in a time where we have to start thinking about where do we start needing to be thinking about developing biodiversity refugia, and where places might change and where might places buffer the change from global change. And so this seems to be a good candidate, and it's all the more reason to really emphasize protect the buffer zones of Manu National Park, because Manu National Park seems to hold a lot of promise for being a control and reference site in this time. And so to that end, the other thing I'd like to point out is that we had a ton of people working on this project, but what was really awesome about our project that has shown a lot of change in our society over the last 30 years is that we have a lot bigger crew, and the authors on our publication we have a very diverse crew that represents 4 different countries, 4 different generations, which is an axis of diversity that isn't mentioned a lot. We have 4 different, my advisor was on this original paper with his advisor. And so we had 4 different generations of scientists who participated on this project. And is a collaboration between Peruvians, Chileans, Norte Americanos, Guatemaltecos, y Colombianos. And so we had a really diverse crew. And the one last message I would like to say is that I'd like to think that whole crew of diverse people and perspectives really enriched the manuscript that's coming along, and has made it a much better product than it's a synergy that's much greater than the individual parts that are part of it. And so I'd like to think that is one aspect, that a very diverse crew to estimate at a very diverse place has led to a really rich and exciting study. And we're looking forward to a lot of interesting collaborations in the future. So that is all I have for you. And with that, I'll take any questions.
So I do have a question for you, Ari.
Yes.
So you said that you haven't seen a lot of change over the past, 30 years because the climate is pretty much very similar since then. Do you think that you'll see a change with climate change
in the future?
Yeah.
I don't know. Don't know what the answer to that question is. Apparently, the idea here is that because Manu is situated at the foothill of the Andes, the accumulation of clouds that creates precipitation pattern are in between two areas of two larger regional. It's sandwiched between two larger regional areas that are experiencing fluctuations in climate, but that this region is kind of in between these two areas. And so that's the only thing we can come to say right now. That's the only reason we haven't seen much change. And the extrapolation from precipitation patterns from the nearest location that we have that is measured over 30 years suggests that there haven't been many changes. Whether that will change in the future, I don't know. Don't know the answer. Yeah.
Any other questions for Ari?
I'm surprised Ben Ewing is not doing a big day.
Okay, I think I might have for the mist nets that you were using to catch the birds. How long do you leave them out there for?
So you leave them. Out there for the standardized mist netting that they did. It was sunrise to sundown, or about an hour before sundown, and they did that for 3 consecutive days, and then they ship, and then they move to a new location. Birds get familiar with areas, and so yeah, you don't leave them for longer than 2 or 3 days in one location.
Okay.
Yeah. Oh, there's 2 in the chat.
Okay. Yeah. So Daniela said, "Thank you so much for the talk."
Oh, no, Ben did it. Well, so Ben already went and did his birding. Great. Awesome. Sure thing, Daniela. Glad you can make it.
Any other questions for Ari?
How long was the research, the duration of the project?
Yeah, it was so this was 3 months, but then we did some follow-up the following year for following the flocks, because that takes a lot more time, where we did another couple months after that. So total about 5 months of field work. And we could do it for a lot less time. The original project had at least 2 years of accumulated data from my advisor who had been studying the colonies of Oropendulus and Caciques, and from Charlie Munn, who was another person in the lab who had been following flocks for like a year. But we kind of fast-tracked it because a lot of us had done work on flocks before. So we could literally have an entire crew go in and power through and crunch that into 6 months, and then do a lot of the other work, have a lot more double up, and have larger crews do it for a shorter amount of time to get the same amount of effort. So about 5 months of field work in total. But the amount of time for the analysis has been ongoing. We've had this paper in review for like a couple years now, it seems. At least a year and a half, and we've gone through like 4 major revisions. We're getting a lot of pushback. So that aspect of the work, which is Jose Miguel Ponciano, Juan Pablo Gomez, who've been really leading the statistical part of it, that's been going on like at least a year. Like they've been doing a lot of crunching, and it's been a lot of back and forth with reviewers.
Okay, Cody does have a question. He said,
yes.
When you record birds, how long does it take to go through the recordings and to identify them?
Yeah, it's a great question. So I should clarify that we just have notebooks, and we literally have distance and direction. We have a spatial reference point, and we just note it as we hear it. The only time we're recording is if we're unsure of things in this case. And like, what is that? You record it, and then people will have all their unknown recordings, and then they'll go through and go through them in the lab. And so, but other than that, it's literally just going through and seeing what you hear, and just like, okay, there's Iphrynchus guttatus, 10 meters, 20 degrees. Oh, there's Remorbus myothrinus, 15 meters, 20 degrees. And what you have to do is walk at a constant rate as you're walking along, and not get bogged down in one place for too long, so that you're kind of doing an even coverage of each route. Yeah, but you're literally walking and doing this with a compass as you go along each trail, and each person has a designated trail for that morning. Yeah.
Jessica has a question. She says, were you surprised to find such similar compositions over time, or were you expecting to see that based on the low fluctuations in precipitation and remote location?
Yeah, that's a good question. We didn't know what to expect for the we didn't know what to expect for the precipitation. We were lucky to get anything because there hasn't been consistent reads at the actual station itself. In terms of the community through time, we didn't quite know what to expect, except for the flocks. We've done previous work on flocks where we have actually seen in the mixed species flocks in one area that the distribution of flocks hasn't changed. For instance, in French Guiana, for some past work that we've done, the flock showed up in the same, they have the same territories over 20 years if there haven't been large scale changes to the habitat. So we were expecting the flocks not to change, and we did in fact see the same thing with understory flocks. One thing I glossed over is that we did see changes. There are things like canopy flocks. That is one of the things that did change quite a bit. We saw only 1 of 5 canopy flocks where they had 5 permanent canopy flocks in 1982, and in 2018 we only found 1. And we're not sure what the reason is for that, but the understory flocks were literally in the same spot. Yeah, it's very crazy. So in the previous study where we expected them, they showed the same positioning over 20 years. Now it's almost 30, 35. Yeah. So as long as there isn't large scale changes to the habitat, we had suspected that a lot of the birds that eat insects, the insectivores, wouldn't change. And that is what we found. But the fact that we saw it on such a large scale with everything else was kind of surprising. That was for a lot of the other things. So that was kind of neat. Yeah. Pretty crazy.
I have another question.
Sure.
In your comparison of the 2 studies that was done a good over 50 years or so, did you see any new flocks establish territory? Oh, sure.
That's all right.
Give me a second. I'm getting a phone call. I'm going to.
Yeah, no worries. We didn't see any new flock. So there are no new flocks. There are people who did say that there were smaller, like in areas where there were tree falls. They saw some things starting to come together, kind of in between areas. But there's some evidence that there might be some smaller new things coming up. And then I would say that that's the main thing we'd say. But in terms of new flocks within these territories that were already established, no. But where there were areas on the edge of the plot where you saw some tree falls, there were little groups of things hanging together that some colleagues say that were kind of the beginning of newer types of flocks trying to kind of force their way in, because the area is saturated. Where you have flocks, it's not patchy. Flocks tend to saturate. Like if there's forest, there's going to be a flock there. So you have to have real estate open up for something to show up is typically what you need to see.
That's very interesting. Thank you.
Yeah, sure. Yeah.
Any other questions for Ari? Okay. I guess there's no more questions.
Okay, great.
It was a very interesting talk, and to see like the comparisons between 30 years ago till now.
Yeah.
And like it's also pretty cool to see like all the different birds that you see out there.
Yeah. Great.
Oh, I think. Oh, sorry, I do have one more question because now that I remembered, because I know you said like for like the mist nettings, you guys banned them. Did you guys notice that you guys like recaptured some of the same birds?
We did not. Yeah, we didn't
Interesting.
recapture any. We didn't recapture birds like that were banned a long time ago. No, we did not. Yeah.
Dang,
Yeah.
that's a bummer. That would have been pretty cool to see if you guys did.
Yeah, it would have been pretty amazing, but we did not actually. So yeah, it was cool, but it was still fun. Yeah.
Okay.
All
Okay,
right.
so I guess with that, thank you guys for joining for today's talk with Ari. It's definitely a great pleasure to have him talk about his research. And I hope I get to see you guys on our next expert series talk.
Well, great. Thank you, everybody. All right. Awesome. Yeah.
Bye.
Bye-bye.